In this video, we're going to look at the disaccharides that you need to know. We're going to look at the test for reducing sugars. We're going to compare the structures and functions of starch, glycogen and cellulose, and we're going to look at the test for starch. Having previously looked at the structure of monosaccharides, we're now going to take a look at the disaccharides that you need to know. Disaccharides are created when two monosaccharides are joined together. They are joined together in a condensation reaction to form a glycosidic bond, and a condensation reaction involves the loss of a water molecule. Now, there are three main disaccharides that you need to know. They are maltose, sucrose and lactose. First up, maltose. Maltose is a disaccharide made up of two molecules of glucose. Then we have sucrose. Sucrose is a disaccharide made up of a molecule of glucose and a molecule of fructose. And finally, we have lactose. Lactose is a disaccharide made up of a molecule of glucose and a molecule of galactose. Now let's have a look in this table just so we can compare. We've got maltose made up of two molecules of glucose, sucrose made up of a molecule of glucose and fructose, and lactose made up of a molecule of glucose and galactose. Now, it's really clear here, all three of them have a molecule of glucose in their mix. So the others that you really just kind of need to remember is which has fructose and which has galactose. Let's have a look at how some of these disaccharides are formed. They're formed from a condensation reaction between two monosaccharides. Here we have two alpha glucose molecules, and we're going to join them together to create a maltose molecule. That happens when we remove the water, so the OH from here and the H from here. That allows us to create a water molecule, and our oxygen bridges the gap between our two molecules. So let's see how that works. So the hydrogen and the oxygen move over and they create the water, and our oxygen molecule here forms two bonds, one with the carbon on each side. Okay, now this is a 1-to-4 glycosidic link. I'm going to explain that by numbering our carbons. So let's go back and have a look at how we can number our carbons. So we start all the way over here. Okay, so we're going to number this carbon number 1, and we follow all the way around, numbering this carbon number 2. This one is going to be number 3, and then when we get up here, we can number this one number 4. We do the same on this side, so we're going to number this one number 1. This one down here can be number 2, all the way across to number 3, and back up to number 4. Now, we do also have 5 and 6, so let's put those in as well. So we've got 5 here and 6 at the top. See the same on the other side. So we're going to have 5 here and we're going to have 6 at the top. So if we now redo our bond, we can see that it happens between carbon 1 and carbon 4. Now, there are alternatives. If we're having a look at a glycogen polysaccharide, for example, we could also have 1-to-6 glycosidic links. But in this case, we are just going to have our 1-to-4 because it's forming here, okay, between carbon 1 and carbon 4. So they are 1-to-4 glycosidic links, whereas we could also have 1-to-6 glycosidic links. Some disaccharides, such as sucrose, are non-reducing sugars. So this means they're unable to donate electrons in the same way that reducing sugars do. Therefore, there would be a negative result for a Benedict's test, okay? That's where that Benedict's solution stays that lovely blue colour. There is a test that you can do for non-reducing sugars that still involves the Benedict's test, but you first have to hydrolyse your disaccharides back into their monosaccharides. So let's go over to the lab and take a look. Now that we're in the lab, we're going to have a look at the test for non-reducing sugars. This always starts by making sure you have a negative result from your normal Benedict's test. That means your Benedict's test staying this lovely blue colour, okay, a negative result. We are going to test sucrose. So you take a fresh test tube and a fresh sample, put it into your test tube, and we are going to do acid hydrolysis. So we're going to add some hydrochloric acid to our sample, and we are going to heat it. So I'm going to place it into this boiling water bath over here, and I'm going to start my timer. Now that 5 minutes is up, we are going to do the next step. So we take our sample out of the water bath and we now need to neutralize it, okay? Benedict's only works in neutral conditions. So we're going to take some sodium hydroxide, and the key here is to add equal parts of your sodium hydroxide to your sample. Now, you need to make sure that this is neutral. So you would use some litmus paper or some universal indicator in order to make sure that it is neutral. You then can add your Benedicts to it. And again, equal parts. So we are going to add some Benedict solution in here. Now again, it needs to be heated. So it's going back into the water bath for 5 minutes and we will see the result. So I'm going to start my timer again for 5 minutes and we will come back afterwards. Now that we have reached 5 minutes, let's have a look at the result. So if we take this out from the water bath, we can see that there is this lovely brick red colour showing us a positive result for the Benedict's test. This shows us that the sucrose has been hydrolyzed back into its monosaccharides glucose because it's now giving us a positive result of this lovely brick red colour for the Benedict's test. So your test for your non-reducing sugars is to start off with a negative Benedict's test, to use acid hydrolysis to break down your disaccharide, and then repeat the Benedict's test in order to get your positive brick red result. Now that we're back from the lab, let's analyse the results. So, a test for a non-reducing sugar first involves a negative result for the Benedict's test, okay? That's where it stays this blue colour, okay? Next, use that process of acid hydrolysis to hydrolyse your disaccharide, so break it down back into its monosaccharide components. Then, after making sure your solution has been neutralised, you then repeat your Benedict's test with a fresh sample. If you then get this lovely red result, okay, so this brick red precipitate forming, it shows that your original sample was a non-reducing sugar, as it's now been broken back down into its constituent monosaccharides. So you still use your Benedict's test, you just use it in a slightly different way. You need to hydrolyse those disaccharides into monosaccharides in order to gain a positive result. Now that we've looked at the disaccharides, let's move on and have a look at polysaccharides. Polysaccharides are simply polymers because they are made of repeating monomers. In this case, they are made of repeating monosaccharides. There are 3 main polysaccharides that you need to know about: starch and glycogen, because they are both polysaccharides of alpha glucose, and cellulose, because that's a polysaccharide of beta glucose. So let's start with the structure and function of starch. So this is our large starch molecule. You can see that it is coiled as well as branched. So let's have a look at it in some more detail. So starch is found in small granules in plant cells. It's a polysaccharide containing alpha glucose. They are joined with 1 to 4 and 1 to 6 glycosidic bonds, meaning that it's branched, and it's also got a helical or coiled structure. So your 1 to 4 are going to be kind of around the loop, and your 1 to 6 causes the branching to occur. So let's compare structure with function. So it's a polymer of alpha glucose. This means when the glucose is hydrolyzed, it's easily transported and it can act as a respiratory substrate. Okay, it's branched, so it has both 1 to 4 and 1 to 6 glycosidic bonds. So because it's branched, it means that there are more ends available for the hydrolysis by enzymes. Okay, so those glucose molecules can be broken off quite easily. It's insoluble, okay, so it doesn't dissolve in water, meaning it does not affect the water potential inside the cells. It also means that it can't easily diffuse out of cells. Okay, and finally, it has this helical and coiled structure. This means you can store quite a lot of it in a small space because it's quite compact when it coils up. Now, an exam technique for you. When you are writing about this in an exam answer question, it's important that you use language that allows you to link your structure and your function. Okay, so that is a really good phrase to use. You can say things like, it's a polymer of alpha glucose so that the glucose can be easily transported and can act as a respiratory substrate. So let's go over to the lab now so that we can have a look for the test for starch. Now that we're in the lab, we're going to have a look at how we can test for starch. We have two solutions here today that we're going to test. We've got some starch and we've got some sucrose so that we can show you both a negative and a positive result. Now this test is really straightforward. We simply take some of our solution, so we're going to start with our starch, we put it into a test tube, and then we add some iodine, because iodine is the chemical that we use to test for whether starch is present. So we just add a few drops, and immediately you can see that the solution has gone blue-black. So let's now have a look at our sucrose. So if we add some sucrose here, this time when we add our iodine, we are hoping for a negative result. So again, a few drops of iodine in here, and you can see that this time it has stayed this red-brown colour. Okay, so this is our negative result. So if we compare, we have our positive result, which was from our starch, that's blue-black, and we have our negative result from our sucrose, which is staying that lovely red-brown colour. Now that we've had a look at the test for starch, let's summarise. So in order to do the test for starch, you should ideally crush your food sample with some water first. You'll then add iodine or potassium iodide, and a positive result turns that solution blue-black, with a negative result meaning that solution stays the same colour, which is red-brown, or you can say yellow-brown. Okay, so the test for starch is used to test for the presence of starch in samples. Ideally crush it, add your iodine or potassium iodide, a positive result will go blue-black. Now that we've looked at our structure of starch, let's move on and have a look at the structure of glycogen. As you can see from our diagram here, it is more coiled and more branched than our starch molecule. Okay, so it is still a polymer of alpha glucose, and its structure is very similar to starch, but as you can see, it is more highly branched. Unlike starch, glycogen is found in animal cells and bacteria cells, and it's stored in granules, mostly found in the muscles and in the liver. So let's compare the structure and function. It's a polymer of alpha glucose, so it's glucose when it's hydrolyzed off, can be easily transported and still acts as a respiratory substrate. It's branched, so it still has those 1 to 4 and 1 to 6 glycosidic bonds, but in this case, it's more branched, okay? So we need faster hydrolysis by the enzymes, because usually animals have a higher metabolic rate, so they need that respiratory substrate quicker. It's insoluble still, so it still does not affect water potential inside cells and does not easily diffuse out. And it's still helical and coiled, so it's still compact and it can still be stored in a small space. And same again, this exam tip, we still want to be using language like so that, okay? So you can say things like it's branched, it has 1 to 4 glycosidic bonds, so that there are more ends available for the faster hydrolysis by the enzymes. The final polysaccharide that we need to have a look at is cellulose. Now you can see from this diagram here that it is different to our starch and our glycogen molecules. Here we have long, straight, parallel, unbranched chains, and they also have hydrogen bonds between the adjacent chains. So in detail, cellulose is the main component of plant cell walls. It provides structure and strength and prevents the cell from bursting due to osmotic pressure. It's a polymer of beta glucose. Okay, so remember our starch and our glycogen were alpha glucose. This time we have beta glucose. Therefore, we have unbranched straight parallel chains as alternate molecules are flipped through 180 degrees. So let's have a look at how that works. Put up a beta glucose molecule, we can add a second beta glucose molecule that we can rotate through 180 degrees and add a final beta glucose molecule. Now you've got to imagine this would be a really long chain. They join together with our condensation reactions, okay, removing the water. Our oxygen then up here is going to join to the carbons, and it has flipped down there and it is going to join to the carbons. Okay, now between our chains we have hydrogen bonds forming, okay? So they're going to form between the hydrogen and the oxygen molecules. Now, hydrogen bonds individually are quite weak, but when you add them all together across your whole cellulose molecule, they become quite strong. So the straight chains of cellulose run parallel to each other, with the hydrogen bonds forming between the chains. They form cross linkages. So as I said, individually these hydrogen bonds are quite weak, but cumulatively they provide strength okay, to that cellulose molecule, allowing it to provide structure and strength to the plant cell walls. These chains are known as microfibrils. So let's have a look at structure versus function. So it's a polymer of beta-glucose, so it only has 1 to 4 glycosidic bonds, so it forms long, straight, unbranched parallel chains. There are hydrogen bonds that form between these chains, forming cross-links, creating microfibrils, and they provide strength. Now, you will possibly see in diagrams and textbooks that these microfibrils can also join together and form fibrils. That's fine, okay? So microfibrils provide strength. So another exam tip, okay, again, we are using language that says so that. So it's a polymer of beta-glucose, 1 to 4 glycosidic bonds, so that we can form long, straight, unbranched parallel chains. So in that video, we have looked at the structure of disaccharides, the test for non-reducing sugars, the structure of polysaccharides, and the test for starch. In the next video, we'll be looking at the structure of lipids.